about protein synthesis or protein production today. And we're going to go through two different processes. We're going to start with transcription, and then we're going to go on and talk about translation. Okay, so transcription. Basically, in a nutshell, what we're doing here is we're going to copy a single gene. So remember we talked about yesterday that the gene is part of DNA. It is the code that gives instructions, kind of like a blueprint, to tell our cell how to build a certain protein. That's actually, this is the district attorney's office, and they have a case that we're going to be working this weekend with the Bloodhound, so had to take that. All right, so let's get back to this. We are talking about transcription. So this is making a copy of a gene. And the reason we need to make a copy is because remember I told you that DNA, those 23 pair of DNA, are really rather big. If you were to take each of the pair of DNA and line them up end to end, they're about a ½ an inch long. They're about 1 centimeter long. So that DNA is wound really tightly and compressed like a coil into the nucleus of our cell, which, for instance, let's think about a red blood cell that we saw yesterday under the microscope. The diameter of that red blood cell is about 7 micrometers. And then you take other cells in the body which are even smaller than that, and you've got to push that DNA into the nucleus of that cell. If you were to take that DNA out of the nucleus, it wouldn't even fit in the cell. It would be like a spring coming uncoiled. You would destroy the cell. So we have to just take like a photocopy of one part of the DNA so that it'll fit through those little nuclear pores into the cytoplasm where the little worker bees are located that actually do the physical building of this protein. Now remember, we talked about the fact that proteins are amino acids that are all bonded together. And what's the name of that bond between the amino acids? A peptide bond. So we're going to be forming a polypeptide. And to start this whole process, you have to have something that binds to the DNA and says, okay, so here's the gene I want to make a copy of. And there's several different chemicals that can do this, but we've already talked about one really important one, which is cyclic AMP. And we can have cyclic AMP come and bind right before that region that has all those A's and G's and T's and C's. And yesterday I didn't tell you this, but this region where it's the beginning of our gene, they actually refer to this as the ta-ta box. Now, that's not what you think it is. It just means a lot of A's and T's, followed by our G's and C's. This is the ta-ta box. Okay? Now, where our cyclic AMP binds is right before this box in a region that's referred to as the promoter site. So this is an area right before our box, and our cyclic AMP is going to bind there. Now, other chemicals potentially could bind there as well. For instance, some of our steroid hormones, testosterone, estrogen, cortisol, DHEA, these are all different steroid hormones we have. They can all also trigger the process of protein synthesis. So cyclic AMP is going to bind to our promoter site. Attract an enzyme that is going to do most of the heavy lifting here. This enzyme is called RNA polymerase. So RNA polymerase will bind at the beginning of our gene.
Is that after all the A's, the G's,
It's binding right at this ta-ta box, and then it will begin its job at the gene. Now the first job of RNA polymerase is it's going to go down our DNA, and it's actually from the beginning of the gene only as far as the end of the gene. It's going to break all the hydrogen bonds between our base pairs. So that means as the DNA extends out here, okay, further, it doesn't break the hydrogen bonds towards the next gene.
So it's like a zipper?
Yep, exactly. So like a zipper, what this is going to do is this is going to prevent the two sides of DNA from holding on to each other anymore. And what we're going to see is the two sides move apart. And what this does for us is this gives us a little bit of room to work in order to build this copy, being able to move the two sides. Now RNA polymerase is going to come along and it's going to build a complementary copy of one of our strands of DNA. And just like we read it 5' to 3', so is RNA polymerase. So it's going to go down the copy 5' to 3', building a complementary strand. Now when it does, there's going to be a few differences. First of all, in the backbone of this complementary copy, we're not going to have deoxyribose sugar. We're just going to have ribose sugar, so that's a little bit different. So it'll be phosphate, ribose sugar on this backbone, okay? So there's one of the differences, two really. We're only making a single strand copy, not a double strand like DNA. And we're going to put ribose sugar in this backbone instead of deoxyribose sugar. The next difference is, instead of thymine as a nitrogen base, we're going to use uracil. Okay, so this is going to be a U instead of a T. And then, of course, this will be A, C, U, 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 G, A, G, 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 A, U, C, C, G. Thank you. Can't write. So we're using uracil in place of thymine. Okay, so let's go over this from the top. We're going to start out with cyclic AMP binding to our promoter site. By the way, which enzyme is it that converts ATP to cyclic AMP? Adenylate cyclase. Excellent. So we have cyclic AMP binding to our promoter site, and that attracts RNA polymerase to the beginning of our gene at our TATA box. First job of RNA polymerase is to go through the 2 DNA strands and break all the hydrogen bonds between the nitrogen base pairs. This causes the gene to separate. Those 2 strands are bubbling out, you might say. Now, RNA polymerase has a little bit more room to work. First thing it's going to do is build this backbone using phosphate and ribose sugar instead of phosphate and deoxyribose sugar. Then we're going to substitute uracil for every place that thymine should be. And now we have what we would refer to as pre-messenger RNA. Not quite done, can't call this messenger RNA yet, so we're going to call it pre, because we've got to do a few other tweaks to it before we can send it through the nuclear pore out into the nucleus. Any questions? Okay, so let me erase a little bit here. Wait, no, okay. Oh, I should have asked. Any pictures? All right, never mind. Oh, okay, no problem. No problem. Okay, so we really don't need this DNA anymore because we already made the copy of the gene. Now all we have to do is alter our pre-messenger RNA so it can get ready to take its trip. So the next thing that RNA polymerase is going to do is it has to do something to let the different organelles in the cytoplasm know which end is which. Which end do we start with, which is the beginning, which is the tail. So RNA polymerase is going to add to the beginning end what is referred to as a methylated cap. And this is a chemical structure that has a lot of methyl groups in it, and so that's what they refer to it. So we know now when this is sent into the cytoplasm that the end with the methylated cap is the end to begin with. The other end, in order to know that it's the end, RNA polymerase will also add a whole bunch of adenines. And we call this the poly-A tail. So now our organelles are going to know the beginning from the end. While this is going on, there's also going to be some editing of our pre-messenger RNA. So the RNA polymerase is adding the cap and adding the tail, but it also has to do some editing because this code is not exactly right. So the RNA polymerase has to go through and kind of proofread. You know, when you write an essay, you don't do it perfectly the first time. You got to proofread it, make sure there's no spelling errors, that kind of thing. So that's what RNA polymerase is going to do. It's going to proofread this code. And as it goes along, our pre-messenger RNA strand is going to go, oh yeah, I knew that wasn't supposed to be there, and take that out. And continue to find sections of our messenger RNA that should not be there. So it's going to literally act like biological scissors and cut out, or what we would call splice out, this bad code. The code that we're going to cut out is referred to as an intron. So this would be intron 1. This code here would be intron 2. This would be intron 3, and so on. These are the codes that don't belong.
So the others would be the exons?
Yes. So now, once we splice this up into pieces, we got to glue it back together. And we say when it's glued back together, we've annealed it back together. The good stuff that's all going to be glued back together, the stuff that remains, we call those exons. So this portion would be exon 1, exon 2, exon 3, exon 4, and so on. Now remember, our genes in reality are much, much larger than what I've drawn on the board here. Now, when all of these exons are annealed or glued together, you have your finished product. This is going to be an mRNA strand. So the finished product obviously is going to be a lot smaller.
Is the RNA enzyme, does that just like start where the beginning of it is and then it just...
Always
so,
reads 5' to 3'.
So the methylated cap is attached to that or?
Yes.
Or
To the RNA polymerase? Or to,
to the chain? But
To the chain.
The RNA polymerase is attached to the methylated cap?
no, the RNA polymerase is reading all on its own. It's outside of it.
But it's just still like somewhere attached on there somewhere?
Yes.
Okay.
Yes. Any other questions?
So the 48 gel is only made of adenine?
Yes, only acting. Mhm.
Thank you.
You're welcome. Anything else? Realize this is a potential essay, right?
Yes.
Okay, yeah.
In regards to the poly A tail, is there a specific number of adenines that?
Not that I'm aware of.
So it's already?
This is now mRNA. Yep, this is our finished product.
So this is still happening within the nucleus?
This is all in the nucleus, yes. Transcription is all in the nucleus.
So it's just in the intracellular fluid? Has it passed through?
In the nuclear fluid?
Yeah, the nuclear fluid?
Mhm.
Okay.
Any other questions?
Do they ever, are those introns ever replaced by something?
No, no,
So
they're
how
not.
do, oh, okay.
Okay,
And
so-
nothing's ever gonna reattach back to the initial DNA?
No. No. But where these go, once they're cut out, we don't waste them. Remember I told you about the nucleolus? It's like a storage closet. It's where we get all these nitrogen bases and ribose sugars. It's stored in that nucleolus. So I cut this, C out and I cut some ribose sugar out. We're gonna go store that in the nucleolus so that when we make this again the next time, we'll have these available.
Well, if it's available, it's still the same bad thing.
Only if we're gonna build the same gene.
Oh.
Now, it may not be a problem for the next one. I mean, we have to put this in there the first time, and then we're gonna cut it out. Because the DNA has the wrong code. And so this is what's gonna happen. There's going to be errors in the code, and we're gonna proofread it to make sure the errors are gone. Now, sometimes, which is kind of interesting, is that when we slice this up into sections, and you have exon 1, exon 2, exon 57, whatever, you can actually have what they call jumping genes. And so instead of putting exon 1, and then exon 2, and then exon 3, when they're all separate like that, you may have exon 1 jump to past exon 64. And then it'll start with exon 2, and then 3, and then 4. And maybe 4 will jump to 27.
How's that happen?
I have no idea. But what it does is it gives you a different product, doesn't it?
Yeah.
If you mix the code up, you have a completely different protein that you're going to build from this code. So if I took 1 and put it after 3, I'd have a totally different code, which when my organelles read this to build the protein, I build a totally different protein. So it gives us an advantage in that the code isn't just stable and this is all you can do. If you mix this code up, you can actually build plenty more proteins. So when scientists were starting to learn this, they're like, oh great, if we just learn this code, we'll be all great. And then they found there were jumping genes. And somebody won the Nobel Prize for that, but it sure made life a little bit harder to figure out how we build every protein in the body. Alright, so when scientists were starting to study messenger RNA, they started looking at all the bases on messenger RNA, and they figured out that there was definitely a code going on here. And they found that 3 bases grouped together on messenger RNA would be the code for a single amino acid. So if you had UUU grouped, you'd have what we call phenylalanine. Or maybe if you had CCU grouped together, you'd have what we call proline. Now, they named these 3 nitrogen bases grouped together a codon. So we have multiple codons here. And if we were to use this chart, we could figure out which protein messenger RNA wanted us to build. So for instance, AUG, if you look on the chart, this is the code for the amino acid MET, or methionine. It's also the start code. That's because on basically every messenger RNA, AUG is the first codon, which also means that methionine is always the first amino acid. So what's the amino acid for GGG? GLY, that stands for glycine. And what about CAU? His, which is histidine. And UUU? Phenylalanine. And CGG? Arginine. And GAA? Glutamine. And AAA? ASN? aspartame, and CGC.
Oh, wait.
Wait, that's
AA or
LYS.
LY?
Oh, lysine, excuse me, thank you. I read it wrong. LYS, our lysine, and CGC,
ARG.
that's arginine again. And UAG,
Stop.
this is stop. This means we came to the end. Now obviously our protein would have more amino acids because remember I told you that the amino acids in the human body are between 1,500 and 50, or excuse me, the proteins in the human body are between 1,500 and 50,000 amino acids long. So this would be a very small code.
Would it always start with MET?
Yes,
Always, every
Always.
single, okay.
yes. Yeah. Okay, so now if we saw and we knew what the messenger RNA was in a laboratory, we could build the protein. But inside the cell, what's going to happen to start the process of transcription is this messenger RNA has to leave the cell through the nuclear pore. And as soon as it does, remember I told you that the rough endoplasmic reticulum is attached to the nuclear membrane. So this messenger RNA goes through that little pore and it attaches directly to the rough endoplasmic reticulum, which is like a little conveyor belt. It's going to move our messenger RNA along until it gets to a ribosome. As soon as it gets to the ribosome, remember we talked about the fact ribosomes can separate. They have a large piece and a small piece, or what we call large ribosomal subunit, a small ribosomal subunit. As soon as our rough endoplasmic reticulum moves this messenger RNA along to the ribosome, the small ribosomal subunit will bond to our messenger RNA. thank you so much. Let's just go over what we just went over. By the way, our ribosome is also referred to as rRNA, and the R stands for ribosomal RNA. So we have messenger RNA, we have ribosomal RNA. So our small rRNA, or our small ribosomal subunit, is going to bond to our messenger RNA. Now the next important part here is something that's referred to as tRNA. This is also transfer RNA. So anything that's made out of RNA is made out of guanines and cytosines and adenines and uracil. And this is a transfer RNA. It has kind of a T shape to it here. And you can see all the bases that help to make up this shape. Now, one of the things that's very important is that transfer RNA is floating around in the cytoplasm all the time. And so are amino acids. So you eat food, maybe you have a hamburger, you chew that hamburger up, it's full of protein, your stomach digests that protein, chops it all up into individual amino acids. Those amino acids get into your bloodstream and start floating around, and eventually those amino acids are transported into your cell. Active or passive transport?
Active.
Active, very good. So they're going to be transported into the cell. Now you have a lot of amino acids just floating around in the cytoplasm of your cell, waiting to be used. Transfer RNA has the ability to pick up certain amino acids, okay? So if you were to look at, let's say this is a transfer RNA. On one end of our transfer RNA, we're going to have a particular amino acid. On the other end of the transfer RNA, it's always made up of 3 bases. So let's say this base is U, A, C. Now these 3 bases can be different. They don't always have to be UAC on every transfer RNA. They can be different. But UAC tells us that this particular transfer RNA will always be carrying methionine. So if you look at the other transfer RNA, it's carrying alanine, and its 3 bases down here are A, G, C. So anytime a transfer RNA has the 3 bases A, G, C, it will always be carrying alanine. So each transfer RNA has a different codon type of thing up here where you have the 3 bases together. Now, of course, we've already called these 3 bases together here on messenger RNA a codon. So what we're going to call these 3 bases on transfer RNA instead of a codon, we're going to call them an anticodon. So the anticodon is the code for which amino acid this transfer RNA will pick up in your cytoplasm.
Initially when you said that the methionine is part of that, is it still part of the one that has alanine in it as well, as a start? Or
Say that again, I didn't quite understand what you're
so
saying.
the beginning of this gene is, it starts with methionine,
Right.
and then would that be still the same in the tRNA with the alanine?
No,
So you would have alanine as a start.
no, no.
Can you
I'm
explain
not-
that to me?
I'm not sure I understand what you're saying, so let me just see if I can clear it up. Okay, so this, forget the messenger RNA, forget all that.
Okay.
Doesn't have anything to do with anything I'm saying right here, okay? So you have multiple different types of transfer RNA. You have 20 different types of amino acids. You have 20 different types of transfer RNAs.
Okay.
The type, I mean, is it's different with the anticodon. So you're going to find there are 20 different groups of anticodons on the transfer RNA. And each one of these anticodons will code for an amino acid.
Okay.
That's it.
Okay.
No more, no less.
Okay.
So on that overhead, I'm just showing you that there's a different group of anticodon and it's carrying a different amino acid. That's all. That make sense? Okay.
I'm still stuck on the how messenger RNA still has methionine on it.
The messenger it doesn't have anything on it yet.
Oh, okay.
This is, all we did was we artificially read this and I showed you what the code is supposed to be.
Okay.
That's it. I only artificially did it.
She's gonna go step by step on how it happens.
Okay, so bear with me.
All right.
Okay?
And the transfer RNA is in the cytoplasm,
Yes.
floating in the cytoplasm.
Exactly.
So it has nothing to do with the secret.
Say it again, it has nothing with?
To here, it has nothing to do with this, but it's secret.
Not yet. It's
floating.
totally separate right now. It's going to come together soon, but it's totally separate right now. Yes. Yes. Okay, so do we understand that this anticodon will tell us which amino acid is being carried? Okay, so if it's a different anticodon, it'll be a different amino acid that a transfer RNA would be carrying. Okay, so our small ribosomal subunit has bonded to this messenger RNA. And because it does, that seems to attract a transfer RNA that's floating around in the cytoplasm. So we're going to see a transfer RNA coming in and interacting with our messenger RNA. Now, how I know which this is because the anticodon has to be complementary on our transfer RNA to our codon on our messenger RNA. And this is going to be U, A, C. And we know that this anticodon carries methionine. And this anticodon can bond to our messenger RNA. So any transfer RNA that has a complementary anticodon is going to come in and bond with the codon on our messenger RNA. So again, our small ribosomal subunit has come in and bonded to our messenger RNA. this attracts our first transfer RNA into the area. The anticodon on transfer RNA is complementary to the codon on messenger RNA, so the nitrogen bases will bond. We get some hydrogen bonds going on. Okay, so notice on the overhead, here's our messenger RNA strand. And the first thing that happens is our small ribosomal subunit comes in and bonds. That then attracts our first transfer RNA. Codon and anticodon are complementary, so they bond. This then will attract our large ribosomal subunit, and that's what we see here. This is the large ribosomal subunit. Now there are two hollow regions in our large ribosomal subunit, and our first transfer RNA is sitting in the first hollow region. This area right here is referred to as the P site. The second hollow region is empty, and this region is what we would call the A site. So again, as soon as our transfer RNA bonds to the messenger RNA, a large ribosomal subunit will enter into the picture and put our first transfer RNA into the P site, and our A site is empty, ready for our second transfer RNA to come along. And it doesn't take long before our second transfer RNA is attracted into the A site. Now, two things. Number one, what is the anticodon on this transfer RNA? CCC. Very good. Because it's got to be complementary. So it's C, C, C. Which means that codon and anticodon, they're going to bond. Second, what's the amino acid on this transfer RNA? Glycine. Because remember, we looked at the chart already and found that the code for GGG would be glycine. here is building a protein. So what we have to do is we have to bond amino acids together. So we've got these two amino acids here, and the next step is to form a peptide bond. And that's exactly what happens. We're going to have a peptide bond formed between these two amino acids. Now, keep in mind that originally these amino acids were floating around inside the cytoplasm. And really, if they wanted to, they could bond to each other at any time. But the problem is, you have to bond certain amino acids in a particular order to get the right shaped protein. And we all know that proteins all have a very particular shape. If you get even one amino acid in the wrong order, so for instance, if I were to take this glycine and I were instead to substitute it with the amino acid valine, now my protein is completely in the wrong shape. And this is exactly what happens when people have sickle cell anemia. Now, we know that your red blood cells are supposed to have that biconcave disc shape like you saw under the microscope, right? But in sickle cell anemia, they have one amino acid out of order. And because of that, the protein that helps to make up the shape of the red blood cell doesn't form correctly. And now, instead of that biconcave disc, you get a red blood cell that kind of looks like this. And what happens is they have these points that as they're going through the capillaries, they get stuck in the walls of the capillaries, and they get stuck in each other, and they start to dam up those capillaries, and blood can't flow. And by the way, anytime your tissue has a really low oxygen supply, it gets really ticked off. And it starts releasing chemicals that cause pain. And so your people who have sickle cell anemia, when they have these flare-ups where things get really stuck, they'll be in lots and lots of pain. They have some severe pain because multiple parts of their body are not being oxygenated. And it's a real problem. But simply put, they only have one wrong amino acid. And that's also because they had one wrong base originally in their DNA. They had what we would call a point mutation. Just one base was wrong, and it creates this massively horrible disease. So we have to have the right amino acids in the correct order to form the right structure. So we can't just let them, willy-nilly bind whenever they want inside the cytoplasm. Plus, it takes too long. This allows us to create proteins very, very quickly. Any questions so far?
So what do the amino acids in your cytoplasm that are floating around, what do they do?
Some of them can bind every once in a while, but they usually fall apart too. And they don't usually stay together. Waits till this process occurs.
You said it causes a lot of pain. Would that be linked to something maybe like fibromyalgia or something?
It would be very similar in pain, but it wouldn't be the same disease. Fibromyalgia is a stress-related autoimmune disease. which, by the way, there's a problem in the genetic code also. You can't have an autoimmune disease without having problems in your DNA. Okay, so now, once our second transfer RNA comes in, we have this little teeny 2 amino acids joined together. Now we've got to join a whole bunch more. So the next thing you're going to see is the small and the large ribosome are going to move down 1 codon. So we've got our next transfer RNA ready to come into the A site. So watch. The transfer RNA that was in the A site here, it gets kicked out. It's gone. So this transfer RNA used to be, this transfer RNA, let me go back, I said that backwards. This transfer RNA here, the UAC, this is in the P site, okay? The CCU, this is in the A site. So the UAC that was in the P site gets kicked out. The CCU that was in the A site moves over to the P site, and the A site was empty, so we brought a new transfer RNA in. Okay, so let me draw that, show you again.
Isn't there an E site? Where's the E site from there?
E means exit, so when that guy exited, it just sort of went through the back door and was out.
Okay.
That's all that means. Okay, so we have, well, both of them do it, so let me move this one over too. We have both the small and the large ribosomal subunit moving down 1 codon. So notice our first transfer RNA that was in the P site, it's not in there anymore. It exited, it went out the back door, what they call the E site. The second transfer RNA that was in the A site, it's in the P site now, and the A site is empty. Just long enough for us to bring in another transfer RNA.
So one the P site exit, and then
Yes.
one of the A moves to the P, and the A remains empty, right?
Mm-hmm. That's correct. Probably going to be about 15 minutes.
I don't know if you're okay, but she's video recording.
Okay, so the large and small ribosome has moved down 1 codon. The transfer RNA in the P site has exited. It's not in the ribosome anymore. The transfer RNA that was in the A site is now in the P site. Our A site is empty.
What happens to that peptide bond that's formed?
Not there yet.
Ah.
Hang in there. Patience. That's next.
Nothing in her.
So now, the bonds for this transfer RNA are all going to break. First bond that's going to break, between the codon and anticodon. No more bond. Second bond that's going to break, between the amino acid and its transfer RNA. No more bond. So there's nothing to hold this transfer RNA here. It floats off. It's gone.
Would it ever be recycled
Yes.
and used for another?
Absolutely, it's recycled all the time. So notice, peptide bond, still attached.
So to pick up another MET?
Yes,
Methylene.
only MET. Mhm. Did you need me? You're just here just because you miss me? There's no class?
No, that is class preparation.
Oh, that's right.
So,
Are you guys meeting here today?
no, I'm actually really just trying to do it.
Then you're supposed to.
It's been alright.
Yeah, he gave us a review yesterday, and I figured, he said, look, let's do this 100 class.
And they didn't show?
So I think he can answer every question.
So when they come and complain to me about how it's so hard and everything, I'm gonna send them right to you, and you can smack them. I don't wanna hear it. Well, if you wanna use the table in the back and stuff.
Work has been late, so I'm tired. But I still came.
Good for you. Now Jason was here
Tuesday.
Tuesday.
Yeah.
So maybe you should email him and say that Thursday is the best day for you, because I don't think he knows that. She didn't show up? So since you're here and you're showing up, I think you need to email him and let him know or text him or whatever. And say, hey, look, I really want to meet you.
Yeah, I'll text him.
Yeah, because I think he was really disappointed too. So it would be nice if he had somebody else to shoot things by.
Yeah.
Because he was here for hours waiting for people.
I would on Tuesday.
Text him, I'm telling you, text him. Make sure you get their crap together.
Okay, I'll
Okay?
try.
Yeah, because that's what you're good at. Okay? All right, so let's go back to this. Again, this first transfer RNA, it's gone, it's out of there, it broke all its bonds, and we have an A site that's empty, so we're going to bring in another transfer RNA. Same thing, going to repeat. So what is the anticodon here? G-U-A, exactly. G-U-A, and we're going to bond. And then, what's the amino acid? Histidine. H-I-S. Okay? Next step, what's it going to be?
Both lights and histidine for peptide one.
Exactly. These guys are going to form our next peptide bond.
So the MET is just kind of floating around there?
Uh-huh. It's just waiting. See what happened? We just repeated.
The east side.
our new transfer RNA comes into the A site, codon and anticodon bond, we form a peptide bond between the amino acids. What's going to happen next?
It's gonna move down.
It's going to move down one. Our large and small ribosomal subunit will move down to the next codon. And we'll bring in phenylalanine and form a peptide bond. And then we'll move down to the next codon and bring in arginine and form a peptide bond. And move down to the next codon and bring in glucine and form a peptide, or this is glutamate, sorry, glutamate and form a peptide bond. Okay? So let's look at this all over again. So from the beginning, we have our small ribosomal subunit bonding. That attracts our first transfer RNA. The codon and anticodon bond is carrying our first amino acid. Then we have the large ribosomal subunit coming in, and our first transfer RNA is in the P site. Initially, this A site was empty, but we brought in our second transfer RNA. Anticodon and codon are complementary. They bond. We form a peptide bond between our amino acids. Large and small ribosomal subunit moves down one codon. Our first transfer RNA that was in the P site, out of there. But the amino acids are still bonded to each other, so it doesn't go anywhere. The second transfer RNA that was in the A site is now in the P site, and we brought in a new transfer RNA. Codon and anticodon are complementary, they bond. Our peptide bond is formed between the amino acids, and we move down again. And we keep bringing in more and more transfer RNAs, making a longer and longer chain. Yes,
You
question?
know, since we read it 5 to 3, right, how come it's going the opposite way? The ones that come in. The ones that come in and break off.
Others.
Because you know we read it this way, how come they're coming in this way, the opposite?
That doesn't,
I think it's just
I don't think it really matters.
Yeah, I don't think it actually matters.
The
so.
tRNA, you mean? The tRNA?
Yeah.
I don't think it really matters.
Well, what matters is the ribosome is reading it 5' to 3'.
Yeah.
That's what's doing it. So the way that they're coming in doesn't matter. It's just the way the animation is. Now, eventually what's going to happen is this large and small ribosomal subunit is going to come to the end. And the end is a stop codon. So, for instance, here we have UAG. There are a couple different stop codons, like UAA is one also. There are no transfer RNAs with a complementary codon to a stop codon. We can't bring any transfer RNAs into that A site to bond with the stop codon because we just don't have any. So instead, what happens is we bring in this chemical here that's called a release factor. Okay, now let me tell you about the word factor, because we're going to come across a couple factors over the next couple weeks. In biology, when we use the word factor, it means it's a chemical that we don't know what the heck it is. So we just call it a factor. And then once we figure out what it is, what its structure is, then they give it some other name. So they'll call it like a release hormone or something later on. So anytime you come across the word factor, you can just go, okay, they just don't exactly know what this is yet. But we do know what it does. When that release factor enters into the A site, it makes it all fall apart.
Is the large ribosome still attached?
No, everything falls apart. This one?
Oh, yeah, I just wanted to see if the business stacker had, an anti-codon.
You know, we wouldn't call it an anti-codon yet because they're not really sure. So just say it binds with it. Because they don't know what the chemical structure is yet. Any questions? Okay, one last thing I want to show you. Well, a couple things. This is a little cartoon showing you the same thing we just looked at again, with our transfer RNA coming into the A site and then the P site. And you can see it building the protein as we go along. Okay? Watch it one more time. Kind of gives you a feel for what's going on. Okay, so in this next picture, in blue here, okay, at the top, this is supposed to represent our messenger RNA. And then we have a large and a small ribosomal subunit, and the little dot is an amino acid. Now, as this ribosome moves down and it's building our protein, as soon as there's enough room, another ribosome hops on, and we start building another protein. And as soon as those two move down, another one hops on. And you'll have hundreds of ribosomes going down this one messenger RNA, building multiple proteins. Now, by the way, when we go through the process of transcription and we're making our messenger RNA, we don't ever make just one messenger RNA. When we go through transcription, we will actually make hundreds of messenger RNAs of the same kind, and then those will all move into the cytoplasm at the same time, and we'll be able to make thousands of the same proteins at the same time. So we never make one messenger RNA, use one ribosome, and make one protein. That's not how it works. We're making tons of these all at the same time. Matter of fact, some of our cells, at the peak of their production, they can make 2,000 proteins per minute. It's pretty good. They're going pretty fast. Any questions?
Take that.
On the exam, are you going to be doing transcription with translation in the
Yes.
essay, or is it going to be two separate?
No. Together.
Does that detail that you just said about how they make hundreds need to be in the essay? Or
No.
Okay.
So we have 2 essays,
One of those.
What?
one of those.
Yes. So, since we're talking about the exam, let's do that. Okay? So your exam is going to be made up of 100 multiple choice questions. And again, remember, it's Monday. So we're going to have 100 multiple choice questions, so you have to have that Scantron, 50 on the front, 50 on the back thing, and a pencil. And then I told you that there's going to be some fill in the blanks. And you'll have one of 2 essays. Either explaining the process of the electron transport chain, which will include explaining NAD in massive detail. And then FAD, remember I told you, shorten it up. You don't have to go through the whole thing. Just have to tell me that it goes to succinate, it bypasses 1 pump, only stimulates 2, so we only get 2 ATP. Okay? Or you're going to have transcription and translation together as an essay.
So the first one, with all that, taking that to the protein, making separate.
No.
So after the exams, we have to stay.
After the exam, you can stay.
I mean, we have to.
Are you asking me if after the exam you get the privilege of going home early? Is that what you're, maybe? You're not sure if you want to ask me that or not? The answer to that is yes.
Okay.
Once the exam is over, You're done for the day, because I find it's useless to try to lecture to you after an exam. It just like, it goes off into the ether somewhere, and you're completely not even paying a stitch of attention, so it just doesn't do any good. You don't learn anything.
How long do we have?
How long do you have? Well, if you'd like to take 6 hours to do it, you may. I don't think you will, but you can have the entire class time to take your exam. Now what we'll do is we'll do the multiple choice first, then I will give you a half-hour break, and then you will come back and do the essay.
So we'll start exactly at 8.
Huh?
At 9:30, I'm sorry.
I'm like, I'm not gonna be here at 8. Out of your mind. I on purpose do not start my classes at 8. I used to
I'm
teach
sorry.
classes at 8, and y'all are zombies at 8, and so am I. So no, 9:30.
9:30 was?
Yes, we will be here at 9:30, exactly. Exactly.
So along with the multiple choice, will you do that fill in the blank?
Mhm. Oh, no, I'll separate that.
Okay, cool. So like 2 breaks.
Mhm. No, you will not have 2 breaks, it'll go with your essay portion.
Oh, okay.
Any other questions? Yes?
If the essay is the electron transport chain, do we need to explain glycolysis too?
No, because that's part of your fill in the blank. You don't explain anything but the electron transport chain. Just like I put it on the board where NAD comes into the matrix, that's where you start. Now, remember, detail, 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 detail. And I'm going to write all over your exam, not enough detail. Okay? It's all about the nitty-gritty detail. I want to see how much do you know, how well can you teach it. So y'all better be teaching it this weekend.
I'm gonna drive people insane this meeting.
You can text me, okay, not text me, you can't text me, I'm not giving you my phone number. You can email me though, and very privileged few have my phone number. You can email me. I may not get back to you immediately because I'm going to be flown up to Oregon this weekend, and I'm going to be doing a Bloodhound trail, chasing a bad guy, and so if I don't have reception up in the mountains, I may not be able to get to you for a couple of hours. But I will be coming in and out of mountainous terrain because there's a bad guy held up in Oregon somewhere, and so they've asked me to fly up there tomorrow, and I'll be...
Do they pay for all your travel expenses?
I'm just a teacher, okay? Yeah.
So,
Yeah.
should say right here, please be careful when you're hunting the bad guy.
Yeah. I'm always very careful. I have been hunting bad guys for 26 years, okay?
Please
And
tell me you go in armed
I
with something
always,
besides the dog.
I always go in armed, and I always go in with multiple police officers, so I've hunted many a bad guy, and so. Huh? Yeah, except I don't arrest. I don't arrest them. As soon as we find them, me and my dog go, we're out of here, see you later, while everybody else gets into a fight and arrests, and yeah.
Dogs that bang.
Dr. Harvey, that's why cheaters does nothing to you.
That's right. That's right. That's right. I'm gonna hunt you down with my dog if you cheat. Okay. Exactly.
You can smell.
Yeah. So this
Are you
guy.
the only one that ever handles your dogs?
No.
Can your...
No, I teach all my students how to handle my dogs.
Oh, that's cool.
So if you take my research class with me, I teach everybody how to handle the dogs, and the students actually are the ones who do the research with the dogs. I don't even do it. I just watch.
But I mean, like when you go up there, are you the only one that handles them?
You know, yes. Well, I have other children who can do it, but right now, one of them is going with me, so she may handle it, so we may go, you know, in turn, but we're always together. And it's really, you have to have somebody with you at all times, because the person who's handling the dog is so focused on the dog, you don't see what's going on around you. And I've had friends who they didn't go with a second person when they're out looking for maybe a missing hiker or something, and them and the dog actually went over a cliff. Because the dog has really saggy skin, and so when the head goes down, everything goes down over the eyes, and the dog can't even see where they're going. So that dog was just following the scent and the hiker went over the cliff. So the dog followed the scent over the cliff and pulled the handler over the cliff because the handler wasn't paying attention either because, you know, you're like intimately involved with the dog's butt as you're going down the trail because that's like what you're staring at the whole time. It's just the way it is.
Were they
So,
okay?
no,
No.
all three of them were dead. Dog, handler, and the hiker.
Was that one of your dogs?
No, not one of mine. No. I had that almost happen. We were on Mount Santa Cinta. There was a missing hiker at the top of the mountain. They took the lift up, and we went up and started the trail right away. His car was in the parking lot. We trailed right to the lift, went up on the lift, and the dog just took off, man. She took off like a shot. And I was running, and it was nighttime. And I'm running, which was really stupid of me. And I hear these police officers behind me screaming, "Stop, stop, stop!" And I pulled up on the dog, which was hard. She weighed about 150 pounds, and I'm pulling up on this dog because she wants to keep going. And the guy goes, "You need to slow down because 50 feet up there, it's about a 3,000-foot drop." And the guy had been pushed off. He and his boss had gotten into a fight. And the boss said, "Oh, let me make it up to you. Let's go up to San Jacinto and, you know, we'll have some dinner and some drinks." And they did. And everybody knew that he and the boss were up there. But then they went for a walk and only the boss came back.
Wow.
So,
Did you guys
Push 'em right over.
actually, was he actually prosecuted?
oh yeah,
How was that, like, found out? How was
he admitted to it. The really interesting thing. So when a bloodhound's involved and the bloodhound finds this evidence, people go, "Oh, the bloodhound, okay, I'll admit everything." I even had a guy one time, he robbed Taco Bell, okay, in San Bernardino, robs Taco Bell with a gun. He's got a mask, and they're always really nice, very polite, because they always leave something behind. They pull out their shirt, they pull out their mask, because they think if they change their appearance, nobody will find them. So they take off articles of clothing, they take off their shoes, and it's just like perfect for the dog. So we found his mask, and we trailed him for miles until we got to his apartment. We went down the 215 freeway and finally got to his apartment, because the dogs can follow a car too, because your scent is emanating from a car. So he's up in an attic of his apartment, and so we were able to get him down from there with no problem. And we were in court waiting to testify against this kid, and his defense attorney comes up and she says to him, "Look, the bloodhound found you fair and square, and you know it, so just cop to it, because you're never going to get off." And so he goes, "Okay." I'm like, "You stupid idiot, don't ever cop to anything." And he give it a fair try, you know? And he did. They gave him 19 years. Okay, now, I always remember this story because he was 19 years old. He only got $19.38 from Taco Bell. And he got 19 years in prison for that. And I'm like, that's not right in my opinion. But he used a gun.
armed robbery?
You use a gun in the commission of a crime in California, you are gone, man. So he got 19 years in prison. I actually felt sorry for him, but
How does that work with the dog on the freeway?
Maybe
I mean,
it's
that's
not. Maybe if it's not.
the dog has the ability to, if you could imagine everybody's scent going down the freeway as a different color. So you've got all these colors of the rainbow. The dog is picking out that one color and following it. So they have somehow that talented ability to pick out that one smell with those hundreds or thousands of people and follow it.
Do you guys have to stop track on like certain?
Oh yeah, we've stopped traffic on the 15, the 215. Cops just stop everything and off we go. And the dogs don't know any better because they're the little prima donnas. So we're cruising back and forth the freeway. We're just running down the freeway back and forth. Now we did a case in Barstow where they robbed the Bank of America, and we had to shut down the entire 15 freeway between Barstow and Apple Valley because the dog was trailing. And we finally went to a house right off of Yucca, and the 5 cousins all got together to rob a bank because one of their dads was a doctor in Watts in LA, and he was being prosecuted for tax evasion, and so they decided to rob the bank to help their dad out, or their uncle.
Good kids.
Yeah.
Very careful now.
They were all hiding in the attic too, but that was so obvious because we got into the house and of course they're stupid enough. We say, oh, would you mind letting us with our dog in your house? And everybody goes, oh, sure. Say no, I don't have to let you in, that's my right, no. But they let us all in with a smile because they're just dumb. And you could tell where they had gone up into the attic because there was all these particles of stuff all over the carpet. And so we're walking through and I'm like, and the detective goes, so we all just backed out and then they brought the bite dog in and lifted him up into the attic. You should have heard the screams. It was great. It was great. Because they screamed, hey, get out, we're releasing the dog, we're putting it up in the attic, and they didn't move, so.
So your dogs will just lick them to death?
Oh yeah. Just don't tell anybody that. They think they're really mean and nasty, but they're like, yeah, they're so lovable, it's pathetic.
Dr. Harvey, does law enforcement use this type of dog, Bloodhound? Because have I heard only the German Shepherd, they're only using those.
That's
Bloodhound?
Yes and no. Problem is, sometimes they do, but most of the time they don't.
Yeah, that's right.
Yes, and that's why they're flying into Oregon, because Oregon doesn't have a bloodhound in the state of, they have one bloodhound in the state of Oregon. That's it. Yeah, most of them are the Belgian Malinois. So in the state of California, there are 10 working bloodhounds. And that's it for this entire state.
Yeah, because I never heard of Bloodhound.
But I will tell you, the cool thing about the state of California is, if you have a missing loved one, okay, and you say to the police, after they've done everything they possibly can, if you say to the police, I demand you bring in a bloodhound, by law they have to. The state of California has actually changed the law that says they must bring in a bloodhound if you demand it. Because there was a family in Monterey whose loved one went missing, he was out hunting, they couldn't find him, and the family knew about bloodhounds because they were a hunting family, and so they kept asking, can you get us the bloodhound, get us the bloodhound, and no, they wouldn't do it. And so they paid for a bloodhound, they paid for a friend of mine to go up to Monterey, and they found him in 15 minutes. Now the problem was he had already been there for almost a week, so he was almost dead because it was very cold at the time. He lost multiple fingers on both hands, he lost one of his feet, he lost a kidney, there were multiple other problems that he had, and he only lived for a few years after that because if they had brought the bloodhound in, they would have found him in 15 minutes and he would have been fine. So they did sue, but they didn't actually sue for a lot of money. What they sued was for a change to the law. And they were able to get that, to say that any of us, if they have exhausted all of their resources, they must pay to bring a bloodhound in. So that's pretty cool.
How long does it scent stay around?
Scent will stay around the oldest trail I've ever trailed successfully was 30 years old.
And how far are they able to
The-
like?
The furthest trail I know, no, in a populated area, the furthest trail I know of was 112 miles long. And he successfully, it was a dog named Yogi, and this is in Colorado, and he successfully found a little girl who had been kidnapped. Now, unfortunately, she had been killed too, but her name was Allie. And the parents have started what's called the Allie Foundation, and you can donate to them, and they buy bloodhounds. I've raised a few, but not very many, but they've bought some of my bloodhounds, and then we train them, and then they send them to various law enforcement agencies for free, so that they can have bloodhounds.
Do you like breed bloodhounds also?
I have bred a couple litters, but I'm not breeding anymore. They're so friggin' hard to breed. They're very sensitive dogs. You've got to take care of them like 24/7. I'll leave that to somebody else. But there's quite a few really good breeders in California.
So there would be a possibility that the German Shepherd could not find the girl.
The German Shepherd could never find them.
Could never find.
No, they don't have the olfactory capabilities. Maybe a Labrador. Labradors are very good at it because they're more of a hunting dog. German Shepherds are not. You have to get the right kind of dog for the right kind of job.
Why did this dog named Bloodhound? Because
They do not trail blood, but the reason they name them bloodhounds is because in the 16th century, a Catholic monk started breeding these dogs. He just you know, they were all in genetics, the monks were at the time in France. And he started breeding these 3 different types of dogs. One was from Iraq, it was a white dog. One was from France, which was a black dog. One was from England, which was kind of a tan dog, which is why bloodhounds are usually black and tan with a little white. He breeds these, and then, smart Catholic dude, to make money for his church, he would give them as a gift to royalty all over Europe. And eventually, and they of course would give gifts back to him, and eventually they made it a law that only royalty could own bloodhounds, and so they called them the blooded hound. So they were royal hounds.
Because they have 3 types of blood within it.
Yeah, well, royalty are called, you know, blue bloods, they're special, you know, so these were considered special dogs, they were the blooded hounds. And not until the 1800s were normal people allowed to own bloodhounds. It was only royalty could have bloodhounds.
When did they realize that they were so good at like transcription?
A long time ago.
I don't
A long time ago. As a matter of fact, in England, up until the 1900s, if a bloodhound tracked you and tagged you and said you were it, you didn't even have to go to court. You were automatically guilty. That's how much they trusted those dogs. You were just automatically guilty. They trailed you into your house and said, tag, you're it. That's it, they're gonna hang you.
Do you have to go to court a lot?
Yeah, quite often.
So as an owner, you're certified like that, how it goes, like?
There are no official certifications for bloodhounds. However, there is something called, oh shoot, what is it called? The FBI has it, I don't even remember what it was called. But I was on the board to help to write the unofficial certifications. So yeah, I guess I could say I am, since I wrote the qualifications for how to be a bloodhound handler.
You
And
also train them?
Yes, I also train them.
How long does that usually
I will
take?
look for that.
Training is continuous, but
Start when they're puppies?
you start when they're about 48 days old, so they're about 6 or 7 weeks old, and you continue to train them their whole life. You're always training. It's just constant. You just try to put them into new situations all the time. That's the important thing. So like today, well, tomorrow, not today, this will be interesting because I'm taking a bloodhound with me who, she's really tough. She can handle the terrain, so that's why I picked her, but she's never been on an airplane. So this is gonna be kind of interesting. We'll
So
see
does
how
she actually get to go on the plane with you?
Oh, absolutely, she cannot go on cargo. No. My daughter has been talking to people all morning long. If you want this bad enough, you can, and police associations there, as cheap can come. And she's telling them, if you want this bad enough, you've got to arrange where the dog has room. So it's either gotta be first class or it's gotta be at the head of the bulkhead, just so that she can spread out, because she's a 120-pound dog. And
So I wouldn't trust her cargo on any person. I've heard some stories about.
oh no,
Yeah.
no, never ever. I've flown to different places to pick up new puppies, but I won't put them in cargo. Because they get lost, or they die supposedly, or other workers at the airport just take them away with them. Yeah.
Is it going to wear also his badge?
Yes, she has a badge.
She has a badge.
She has a badge. She has her own paperwork. She has her own ID card with her picture on it. Not kidding you.
Smart.
Did one of your dogs just win an award sometime recently? Because I saw an article in the paper about a bloodhound that did some police work and she won an award.
They
Here
have,
locally.
I don't know if it's the same one.
It was cute. Had a little picture in the paper.
How recently was it?
Within the last 3-4 months.
Oh, no, that's a different bloodhound. San Bernardino Sheriff's Department has gotten a couple bloodhounds, so they want to really push the PR, so they're giving themselves awards. It's really cute. The sheriff is giving the bloodhound an award, you know. Yeah, they're trying to get everybody to understand that they have these dogs, so. Trying to do some good PR for them, which is good.
Can they also get along with other dogs like pit bull, chihuahua?
No. No. And bloodhounds, even though they're very nice dogs, if they get upset with a pit bull, they're gonna win. The pit bull isn't gonna survive. I had a bloodhound who, within 2 bites, she had killed a pit bull. It was the pit bull's fault. It tried to attack her, and she's like, ha ha, not gonna happen. He grabbed her on the neck, but she has a lot of skin, so all he did was grab her skin. She threw him off, turned around and bit his spine and pulled his spine out with one bite. So those dogs are extremely powerful, extremely powerful. But they don't usually fight. They're lovers, not fighters.
Do you have a way to protect your dogs while you're not home? I had a bloodhound in, it was in '96, and it got stolen when we were on vacation.
Yes, I also have an attack dog.
Oh, okay.
I have a German Shepherd, and he is actually there to attack people who go into my yard. Because I had one bloodhound who was shot in my backyard. We had some Hell's Angel problems, and they decided to enter into my yard and shoot one and injure a second. And then so we bought a German Shepherd. And I'm also thinking about getting a Bouvier, because they're going to be even stronger. They're really, really stronger than a Malinois. They're super strong attack dogs. But unlike German Shepherds, Bouviers can be really nice with kids. And German Shepherds are real pissy sometimes. They don't like children as much. They can be very touchy.
I have a question.
Yeah.
So here recently, there's been a lot of mountain lion attacks against dogs, in Zealand and here. Would your dogs be able to handle a mountain lion?
I actually had a bloodhound that was attacked by a mountain lion. She survived. The mountain lion took her face off. Literally, she basically didn't have any face. She swelled so bad that it actually compressed the optic nerve in one of her eyes. She went blind, but she survived.
Would she survive?
Yeah, the entire group of bloodhounds came and attacked it, but she was the first one attacked. They chased it off.
You guys
Wow.
hunt down?
Yeah,
Mhm.
it's pretty bad this year.
Hunt it down? No, you can't hunt mountain lions.
Mountain
Doesn't
lion.
matter what they do to your dogs.
And then
They're,
there's one in Big Bear.
no, they're totally protected.
Yeah.
No,
Yeah,
absolutely
where I
not.
was driving home one night and I saw
I think what happened is the mountain lion
mountain lion.
really didn't know what it was doing and jumped into the backyard. I don't think it meant to hurt anything. We had that happen to a coyote too. I actually felt sorry for that freaking coyote. Those dogs were just, they're like cats. They like to play with their prey. And they were playing with that coyote like you cannot believe. And finally I'm like, leave the thing alone, and opened the door so it could escape. Because it didn't have long to live because they were going to rip its throat out. But boy, they just beat him up. Bloodhounds, they'll pin animals between them, and then the third one will come in and beat it with their shoulder. And it's a very interesting maneuver, but they're a super pack. And they love to run in a pack, and they love to kill in a pack. So,
Wow.
Was that hound and fox and a hound? A bloodhound?
yes, supposed to be a bloodhound. No, it's actually not. We had a little boy up here in Apple Valley that was attacked by some Rottweilers, and he was bit over 1,000 times. He had to go into surgery 4 separate times, and he was, of course, scared to death of dogs. So in order to run this trail, we actually had to have the dog meet the boy, and one of the detectives who was on it, he went over to the kid's house and they watched the fox and the hound together so that he could describe how nice the bloodhound was, and the kid would be willing to meet the dog. So, yeah, it was very sweet.
Did you find out what happened to your dog that passed away?
No, not yet. The report hasn't come back. They're doing a toxicology report right now. I thought she had cancer, but she didn't, so they've sent a toxicology out to Oklahoma State University, I believe it is, to see if maybe she was poisoned.
You say you saw
We had a Mojave Green. Yeah, so that's the other question, to see if she was poisoned.